Okay, part 2 of chapter 16, the pulmonary ventilation. So let's look at some factors that affect pulmonary ventilation, which would be compliance, resistance, and then looking at the clinical significance of some respiratory volumes and a couple of disorders. So let's look at our first topic, which is lung compliance. So compliance is the ease. So the compliance is the same as blood vessel compliance. It's the ease at which your lungs can be stretched. Lungs have a compliance. They're very compliant organs. And so the larger the lung has compliance, it can stretch and you can inhale because of that compliance. And the smaller changes in transpulmonary pressure is needed to bring in a given volume of air because of this compliance, right? So remember, the transpulmonary pressure is a pressure that's allowing your lungs to expand and get bigger. So because your lungs are compliant, you don't really need a huge transpulmonary pressure to increase the lung size. So what are factors that affect compliance in their elasticity and surface tension? So again, this is just compliance. And what is elasticity? Elasticity is the resistance to that stretch, is to recoil. So compliance and elasticity are actually inversely proportional to each other. So when you have more compliance, you have less elasticity. And when you have less compliance, you have more elasticity. So they're sort of opposite forces. Let's take a look at your elasticity here. So I have a couple pictures. This is tuberculosis, and this is emphysema, as you can see. And both of these are going to affect compliance and elasticity. So let's take a look at the elasticity first. So elasticity, again, is this force recoiling. So if you look at what's under less compliant and more elastic is scar tissue. So that is tuberculosis. So when you have tuberculosis, you do have these areas of the lung that become fibrous. Fibrous is tough tissue, it's tough connective tissue, and it's scar tissue, basically. It's scar, and that's why they say scars, right? Scar tissue. So you guys know that if you have a scar on your body, it feels different. It's a harder kind of tissue than the rest of your normal tissue. And so if you have scar buildup in your lungs, such as with things like tuberculosis, or there's another thing called pulmonary fibrosis, or any disease that causes this fibrotic tissue to build up in your lungs, that would reduce the ability for it to stretch. Okay, so it's going to reduce that compliance. And if it doesn't stretch out, your lungs are that force to recoil is going to win, and so that it's going to increase that elasticity. And either in these diseases with tuberculosis or pulmonary fibrosis, it would make it hard to stretch with inhalation, and you would have to, and then the lungs would just constantly be in this recoiled state. So it'd make it hard to breathe. In another example, emphysema, the opposite example is in emphysema, the immune system actually, it starts to break down the healthy tissue of your lungs. It starts to break down the elastin fibers. So it's not as elastic anymore. So it becomes less and less elastic, and it's very floppy. And so you can inhale, someone with an emphysemic lung can take in the air, the lungs stretch out, they're very, very compliant, but there's no recoil because the elasticity is gone. So they can inhale the air, but they can't exhale the air. So they can't, that's the problem with emphysema. So they can't have normal flow of gas exchange because they don't have that expiration. So hopefully that makes sense to you. This is a very scary image of emphysema, right? Look at all of this space that's just destroyed. Another factor that affects compliance is surface tension. And surface tension is inside of the walls of your alveoli. It's watery. There's, our whole body is bathed in water. So there's a fluid layer that lines the alveoli, and that fluid is going to create the surface tension. So this inward, you can see these inward arrows water molecules want to stick to each other, and so these inward arrows are going to show that if you had nothing but water, a film of water inside your alveoli, it would want to collapse. It would want to fold in on itself. So it has a high surface tension. What the lungs produce is a substance called surfactant, right? And surfactant molecules are a detergent, which is kind of funny. So it's like a protein with a lipid component, and it coats the inside of your alveoli, so it reduces that surface tension. So the lungs are less wanting to collapse in on themselves. So the alveoli do not collapse. So the type 2 cells are going to secrete the surfactant, and surfactant's going to increase your lung compliance because it wants to collapse in on itself, right? Compliance is the stretch to expand. And so if you have surfactant, it's going to be easier to expand, and so it's going to increase the compliance. So let's look at, let's see. All right, so let's look at the next slide, which is the resistance. So resistance is pretty much also the same as the cardiac system. So it's created here by inelastic surfaces as well as the diameter. So the diameter is what really is overlapping with the vascular system. You can change the diameter of your airways, the bronchi, the bronchioles, because they have smooth muscle. And so it's called bronchoconstriction and bronchodilation. And that's controlled a couple ways, extrinsically and intrinsically. But before we get started with that, we can see that, all these tubes of your lungs get smaller and smaller and smaller until they reach a very small diameter. And we saw with blood vessels that the smaller the diameter, the more resistance you have. So why is it that the lungs, they have so many small diameter vessels, but actually the resistance is quite low, about 1 millimeter of mercury? And the answer is that there are so many branches of them. So every time you get smaller tubes of airways, every time you branch, every time you get smaller, you're going to have numerous branches. So you're increasing the branches, and so you're keeping that resistance low, even though, right, you're getting smaller and smaller in diameter. Okay. And of course, increasing the resistance is going to make it harder to breathe. It's going to make it harder for that conduction pathway to get air in and out. And so let's take a look at some factors of resistance. So again, we can constrict, right? Bronchoconstriction and bronchodilation. So you can see the images and what that relates to. Bronchodilation is going to be your sympathetic nervous system. So your fight or flight response. You're ready to run. You're wanting all the air to get into your lungs as much as possible. So the sympathetic nervous system is going to dilate your blood vessels. I'm sorry, I keep saying blood vessels, but the bronchioles there, bronchi and bronchioles. And then the parasympathetic system is going to do the opposite, where it's going to constrict them. And there is, so that's your basically extrinsic control. It's the nervous system coming in. There's also hormonal control, so epinephrine, which is also part of this sympathetic nervous system response, right? So if you're under fight or flight and you're secreting epinephrine, the hormone, it can also cause bronchodilation. Intrinsic control, it's basically carbon dioxide. So when you have a buildup of carbon dioxide, that's going to trigger your body to want to get rid of it faster. So it's going to also trigger dilation, right? Because it wants the airways to get bigger, bring more carbon dioxide into the alveoli for gas exchange, for your body to get rid of it. Another one, histamine. Histamine is bronchoconstriction. And if you have wheezing, coughing with allergic reactions, this is histamine. So histamine is, remember that chemical secreted by basophils that is going to mediate allergic responses. In the lungs, it does constrict your blood vessels. It also can increase your mucus secretion. So overall, histamine is going to make it difficult to breathe. And the logic, I think the evolutionary sort of rationale for constricting your blood vessels is to minimize the infiltration of foreign particles into your body. It backfires, right? Because it's very difficult to breathe, and under extreme circumstances, it can become life-threatening. So it's a reaction that we have, but it's not a nice one to have. Okay, so resistance is pretty much done by this constriction and dilation of your bronchi and bronchioles, but also mucus. So mucus does create resistance, because obviously if you have mucus in the way, the airflow is not going to be able to pass by unimpeded, right? It's going to have some kind of block to it. So it's going to increase the resistance the more mucus you have. And the more mucus you have can be due to, right, you are always going to have normal levels of mucus, but then there's illness, allergy, and any disease that can change that. And also, in terms of just the nature of the mucus, that can change too, because cystic fibrosis, remember, you don't have the water coming into the mucus that's produced, and so the mucus is very, very thick and very difficult to move out of the lungs, which is going to make it hard to breathe, increase resistance. All right, next topic is looking at our respiratory volumes and airflow. So let's look at all of this data. The first thing I want to look at is tidal volume, and I think this is the easiest thing to understand is just this normal, relaxed, unforced breath that we have, that we come in and out of our bodies. So that's illustrated here by these sort of smaller waves, right? So the normal volume of inspiration and expiration. Then you have on top of that a reserve volume. So inspiratory reserve volume. After breathing normally, you can still breathe in. So if you sort of breathe in, breathe out normally, and then someone asks you, you breathe in normally, and then before you breathe out, someone asks you to breathe in all the way. So you can actually bring in even more air. And so that's going to be this inspiratory reserve volume. So it's on top. You see how the line, it's on top of your tidal volume. So your tidal volume is here, but you can actually bring in so much more air. That's your reserve. Expiratory reserve, same thing, right? You're at the bottom of your exhale in a normal, relaxed breath, and then you're told to exhale everything you've got. So that is the expiratory reserve volume. Now, the residual volume inside of your lungs is, you're always going to have air in your lungs. You never have completely empty lungs. So it's the air left in your alveoli after exhaling forcefully. So get it all out. Even when you forcefully, you think you can't have any more air to come out, you still have air in your lungs. That is your residual volume. It's important to have residual volume. So RV is residual volume, because it keeps your alveoli from collapsing. There's air always in the alveolar sacs and in the alveoli. And it actually functions to keep it from collapsing. So it's good that it's there. Now let's look at our inspiratory capacity. So we have something called vital capacity. So vital capacity is the maximum volume expired after maximum inspiration. What that means is you breathe in as much as you can possibly breathe in, and then you breathe out as much as you can possibly breathe out. So it's basically as forcefully and as much air as you can move in a possible breath, right? So that's going to go, so we can see that it encompasses everything that we, breathe out with everything we breathe in. So it's a really big, so our vital capacity is this measurement. The functional residual capacity is the volume remaining after the resting tidal volume in our lungs. So our functional residual capacity, okay, is going to be from our normal breath to it basically just includes that residual volume plus what we could have exhaled, but we didn't, because we're just doing sort of tidal breathing. So when we're breathing in and out, we're not using our vital capacity, right? We're not using our entire lungs capacity. So this just tells you it's, what your lungs are capable of holding. So it's your expiratory reserve plus that reserve volume that never goes away. And then the total lung capacity is the volume of air. So here's the total lung capacity. Total lung capacity is the volume of air in your lungs after maximum inspiration. You can see it's just everything, right? After maximum inspiration. So what can your lungs hold? Total lung capacity. What can your lungs hold? Max, right? What's the max? What's the total lung capacity? It's everything from the very, very bottom, all that residual air, all that reserve volume, plus anything possible that you can inhale, right? So it's all of the gas that your lung can hold. So that's total lung capacity. That maxes out. So you can see how this graph goes to 5,700 milliliters. I heard elsewhere that 6,000, like an adult man maxes out at 6 liters. So, it'd be 6,000 mils or 6 liters. So 6,000 would be the mils, be the upper limit. This is sort of an average, normal amount. All right, so that is the things there. Understanding lung volumes, understanding lung capacities. And then we're going to talk about a couple of disorders. There are two main categories of these pulmonary diseases, which is obstructive and restrictive. Obstructive pulmonary diseases are associated with increased airway resistance, so there's something in the way of moving air in and out of the lungs. And it's going to increase the amount of air that's left inside your lungs, right? So letter B, where it says residual volume increases, that should make sense because with every breath, if you're not moving as much air in and out as you can because there's some kind of resistance, then you're not going to have that residual volume, that stuff that's still in your lungs, that's going to increase. And what happens over time is that the alveoli get distended, so they get bigger, which means that the more air, that's why you can hold more air. This residual volume increases because the alveoli they're puffed up with a little bit more air inside of them. The vital capacity also decreases, right? So just how much you can move in and out is not as much because you have resistance. And some examples of major obstructive pulmonary diseases is COPD, which includes things like bronchitis, chronic bronchitis, and emphysema, as you can see here. But asthma is also in this category. In these diseases, what happens is that there's issues, there's increased resistance. So with bronchitis, you can see that those airways are going to be filled with mucus. You're going to have a chronic underlying, a lot of mucus in your lungs and those bronchi. In asthma, the same thing. And emphysema is a different kind of mechanism, but emphysema is also in this category. Oops, sorry about that. So just a quick little, because asthma is so common, I just thought I'd expand on asthma a little bit. So asthma is where the airways are narrow, and it's a response. So asthma is funny because asthma can be caused by many different things. And it's a response that includes narrowing the airways, so bronchoconstriction, as well as making mucus. And the symptoms, because you're having problems moving the air, is coughing or wheezing, shortness of breath, feeling tight-chested. And the causes are variable. So some people have allergy-induced asthma where they're specifically allergic to something, and when they have that reaction, they get that asthmatic wheeze. Sometimes people have asthmatic episodes because of an irritant. So a campfire smoke or some kind of particular pollutant in the air, or ozone. Some people have a temperature. If it's too hot or too cold, it can trigger asthma. Sometimes people have exercise-induced asthma. Sometimes people can get just upset and then they start to have this asthmatic reaction because, again, remember the sympathetic response. Well, this is a little bit because the sympathetic nervous system is supposed to dilate the bronchi. But in this asthmatic reaction, your emotions will constrict the bronchi and you can have an asthmatic attack. So asthma is thought to be this environmental and genetic factor combination of the two. And at the moment, there's no cure, but the most common, I don't have a picture, I was going to put a picture of that, but the most common treatment is an inhaler, right? So it's a little cartridge with albuterol inside, and the albuterol is it's a fast acting, it relaxes the smooth muscle, it's an adrenergic beta-2 receptor and agonist. So it's going to act on the smooth muscles. So beta-2, remember that's an epinephrine, adrenaline receptor. So an agonist is going to mimic epinephrine. And so since adrenaline and epinephrine vasodilate, right, this albuterol also does that. So it mimics epinephrine. It's going to open up those and relax those smooth muscles so the airways can open up and let you to breathe. Okay, and then our next, so those are obstructive pulmonary diseases, increased resistance, and then there's restrictive pulmonary diseases, and this is going to restrict lung expansion. So it's going to be more difficult to expand the lung to make them bigger, and this can be done in a different, I have some examples of this. So the first one is the surfactant issue. So you have increased pressure inside the alveoli. So the alveolar sacs, if you don't have that surfactant, the alveolar sacs are going to collapse on each other. So this respiratory distress syndrome, the alveoli themselves don't want to expand, right? Because they're missing surfactant. So this picture here is this infant respiratory distress syndrome where really, really premature babies can be born without surfactant. And so they need to get surfactant replacement therapy, which is available if you're at a hospital, right? If you can access healthcare. Other things, which I don't have a picture of, are scarring type infections that reduce the compliance. We talked about that with tuberculosis. Or just physically, the bones that you have, the muscles that you have are unable to expand the chest wall. So things like scoliosis, kyphosis, which is where you have a hunchback, those things can actually restrict your lung expansion. So that would be a restrictive pulmonary disease. So restrictive is restricting lung expansion. How do we measure if somebody has obstructive or restrictive pulmonary diseases? So then you need to take these tests, pulmonary function tests, and you do it with a spirometer. So it's called spirometry. So you can see that there's a mechanism where you're going to shut off your nose, you're going to blow into a device, and it's going to measure the amount of air that's coming out of your body. So what they're going to measure is a couple of things. The first is forced vital capacity. So you can see this is basically forced vital capacity with the forced expiratory volume, forced expirate over 1 second. So what's forced vital capacity? This is maximum volume inhalation followed by exhalation as fast as you can. So someone tells you to inhale as much as you can and exhale all of that air as fast as you can. There's no exact time limit here. It just wants you to get all that air out. And then so the only thing left is residual volume. So you're just going to blow out, blow out, blow out, blow out until you have nothing left. And so if you have a low forced vital capacity, you're going to indicate restrictive pulmonary disease, because restriction means that you can't expand the lungs enough to get a lot of volume. So a low forced vital capacity could mean restrictive pulmonary disease. Forced expiratory volume is a percentage that can be, it's a percentage of your forced vital capacity that can be exhaled in a second. So again, the forced vital capacity is how much you can possibly push out in your lungs. You should be able to, a normal forced expiratory volume should be 80% of what you're capable of within 1 second. So in 1 second of forcefully exhaling, a normal person would be able to get out 80% of all of that air minus that residual volume, right? And then we can look at the differences in the numbers. So let's take a look at the graph and see if we can just make sense of the graph. So we have our normal lungs. Here's our normal lungs. And we can see that this is looking at FEV1. So it's that forced expiratory volume in 1 second. And we can see that in normal lungs, the number is here. I mean, the placement here. So at 1 second, look at the volume exhaled. So it goes from here, 0, all the way up. It doesn't really matter, the mils. And then at 1 second, this is where we are. And then in subsequent seconds, right, we reach the total. So the forced vital capacity, you see how it flatlines? That's basically it. We can no longer push any more air out. We've reached the end. The only thing we have left in our lungs is residual volume, which we can't push out. It's always going to be there. So that flat line indicates that's our forced vital capacity. Now, if you look at these other diseases, look at the curves. So I have a little hint here, right? Look at the curves. Look at our restrictive diseases. Restrictive diseases, again, is the lungs can't inflate properly. So the ability to take in air is compromised. So they have a very similar shape to normal, right? But what's the difference is just the amount of air, right? So whereas a normal person can, exhale this amount of air, somebody with restrictive diseases doesn't have that much air in their lungs to begin with. Even though they can still reach the same curve, they can still get out about 80% of that air that they have in their lungs. In that 1 second, the amount of air in their lungs is less. So this shows you a restrictive disease curve, and then we reach full vital capacity as well, a little bit later. But that's the big difference. So in restrictive lung diseases, people have problems getting air in. They don't have a problem getting it out. So that's why we can see the very similar curve with normal. The ratio should be normal. They should be able to get out 80% of the air in a second. However, the actual numbers are lower, right? Smaller, because their vital capacity is smaller. In obstructive diseases, you can see we have a different curve. Obstructive lung diseases, they take a normal breath, and when they're told to fill up their lungs, they exhale as quickly as you can, and that's when they're pushing and pushing all the air out. And you can see that after a second, they can only get out a little bit. They can only get out a little bit of the air. So they've not reached their 80%. And so it takes a while, it takes a much longer time for them to reach that point where they have completely reached that full vital capacity. Anyway, so it just takes them much longer. So the curve is different, and that's indicative of an obstructive disease. So this would be something like asthma, right? It just takes a lot longer to reach that expiration because there's something in the way, right? The airflow is restricted. All right, so let's look at, this is what the book has, very similar though to what I just went over. And then lastly, we're going to turn our attention to alveolar ventilation. So what's going on inside your alveoli? So really we just care about what's going on inside the alveoli, right? Because the alveoli is where gas exchange is happening. So in a minute, so this is minute ventilation, the total volume of air entering and leaving the respiratory system each minute is going to be the tidal volume, what you're breathing in and out, and multiply that by the respiratory rate. So in a minute, how many times are you breathing? So the normal respiration rate is 12 breaths per minute. And the normal tidal volume is about 500 mils of air. And if you do the math, right, that's about 6,000 milliliters per minute. But then, is all that air getting, so 6,000 mils per minute? coming in your body and out your body. Is all that gas getting exchanged? No. So when we're breathing, we're not replacing all the air in our lungs, and actually we're only going to start exchanging 10%. So that fresh air that you just inhaled, we're only going to be able to replace 10% of the air in our lungs with that fresh air that you inhaled. So most of this air is coming into the conducting zone. It's not actually reaching the alveoli. So in this picture, what you see is the fresh air, right? So this is the conducting zone. And this is also referred to as the anatomical dead space, because the anatomical dead space does not exchange gas. So this dead space contains old air that your lungs are pulling, they're still pulling the oxygen out of, but this air is going to just be in your lungs because not all that fresh air is going to replace it. When you inhale, we can see that this anatomical dead space, this old air, is going to get pushed, your fresh air is coming in, and there's going to be mixing of the fresh air and the old air. It's going to lose oxygen, you're going to gain that CO2, and then when you exhale, you're going to push most of that fresh and old air out, right? But you're still going to have some old air left inside of your conducting zone, left inside your alveoli, and that's just dead space. It's not helpful because most of the oxygen's been removed. So that dead space is about 150 mils. Now, let's look at alveolar ventilation, because what we care about is exchanging gases. So alveolar ventilation is going to take your tidal volume and, basically per minute, right? Your tidal volume times the respiratory rate per minute, and you just take away that dead space. And then that's how much air is actually going to get exchanged, right? Because you minus the dead space, and then you see, okay, this is the air we're bringing in. If we take away all that dead air and all the conducting zone space, then this is the air that's actually getting exchanged in the alveoli. So there are some examples here. In your outline, you have sort of a normal baseline. So you have 500 mils per breath at 12 breaths. So that's what we figured out. Normal minute ventilation is going to be 6,000 mils. We're going to subtract that dead space, which is estimated to be 150 mils per breath, times 12 breaths is going to be 1,800 mils. And so about 4,200 mils reach the alveoli. But this is sort of normal. And this is just a quick glimpse at some scenarios here. So if someone's breathing, so remember 12 is your sort of normal breathing rate, 12 breaths per minute. Say someone is breathing at 50 breaths per minute. So they just ran a lot, they just sprinted and they're panting, right? Like just very, very quick breathing. They're not going to be bringing in a lot of air. So their tidal volume is going to be small, right? Their minute ventilation, if you multiply the two, is going to be 5,000 milliliters. But out of that, nothing's reaching the alveoli, because the alveolar ventilation is going to be very, very minimal. If your respiratory rate is 10, so this zero is gotten because we're also subtracting, just so you guys know, subtracting that dead space, the 150 times the minute. The respiratory rate, if you're doing 10 breaths per minute, tidal volume of 500, then you get an alveolar ventilation of about 3,500 mils. And if you're taking very slow, deep breaths in that minute, only 5 breaths per minute, your tidal volume is going to be larger because you're taking deeper breaths, and the minute ventilation is going to be 5,000. But look at the alveolar ventilation, it went up. So you can see that even though we have our minute ventilation is going to be the same, we can see that with deeper breathing, with bringing in a larger tidal volume, that we actually get more alveolar ventilation. So in order to oxygenate your cells, the best way to do that is with slower, deeper breaths. And so that is the mantra that you hear a lot from people who are meditating or who are proponents of like exercise, physiology. They want to oxygenate their cells. It's to take these slow, deep breaths, because that's the most efficient way to sort of get the oxygen to exchange with your blood. And this is just an example. I just pulled this off the internet. Some ways to really do some deep breathing is this diaphragmatic breathing, but I'm not going to go into that. Just one example of there's techniques to change the breathing. And then lastly, just finishing up with an interesting thing. This really doesn't have to do a lot with this chapter, but I just thought it was cool because of COVID. And it is the respiratory system. So I found this paper which says that when you smoke cigarette smoke exposure actually increases the expression of the SARS-CoV-2 receptor. So the virus wants to use this receptor called an ACE2 receptor. So this is drawn in blue here. This is the ACE2. And the virus uses that to enter into your cells to give you that infection. When you don't smoke, right, a non-smoker, you see that there's a couple of ACE2 receptors. When you do smoke, you have more expression. So you increase the number of ACE2 receptors, which means that you are more likely, if you do inhale that virus, the virus has just more receptors to bind to, right? So it can increase the likelihood that you'll be infected if you're a smoker, and it's due to this increase in ACE2 receptors. So I thought that was really interesting to see. This study was done with about, they saw that when they looked at 1,000 or 1,100 people, that people who were COVID positive, 12.3 who were also smokers had to be ventilated or died, and that got COVID. And if you were a non-smoker, the percentage was 4.7. So there's a big difference between smokers and non-smokers in terms of your prognosis with COVID. So best not to smoke during the COVID times or at all, really, right? So review questions. Which device measures volumes of inspired and expired air? This is going to be the spirometer, letter B. Compliance is decreased if lung tissue becomes thickened or there's a lack of surfactant and the muscles have to work harder to breathe. What are two examples of this problem? So this is going to be emphysema and your respiratory distress syndrome. What is the vital capacity? So your vital capacity, remember, is everything that your lung can hold. All right, so this is going to be letter C. Oh, I'm sorry, that's not reserve volume. It's going to be this one, letter B. Tidal volume and inspiratory and expiratory reserve volumes. What are the indicators for obstructive and restrictive pulmonary diseases on a pulmonary function test? Right, if you looked at your choices, it's letter B. Between breaths at rest, some air stays in your lungs. What's the term for that air? Answer is this one, functional residual capacity. How does histamine, a chemical that is released locally during allergic reactions, cause difficulty in breathing? Okay, it is letter C. As with the capillaries in the circulatory system, there is low combined resistance in the conducting zone, despite the tube's decreasing radius. Why is the resistance low? The answer is